Hello everyone, this is Ragani N3. So let us see about the question and answers from the topic sexual reproduction in flowering plants. Okay, so the salient features of this chapter is that we have experienced seen older individuals senescence, undergo senescence and die. So we have this reproduction which guarantees successive generations. Okay. Flowering plants also undergo sexual reproduction and they possess sophisticated reproductive organs including male and female reproductive organs and accessorizing structures. So this chapter will provide you a detailed knowledge on sexual reproduction in plants with detailed knowledge of their structure and mechanics. The mark weightage of the chapter is NEET examination. It carries 3 to 4 questions. In board examinations, the questions are 10 to 14 marks. So distribution of marks. So here we have section A to E. A we have MCQs, B we have 2 marks, C we have 3 marks, D we have 4 marks and E we have 5 mark questions. So number of questions asked in 1 mark is 16. So total mark is 16 and 5 number of questions in section B, 7 number of questions in section C and 2 questions in section B and 3 questions in section E. Okay. The important topics that you have to cover in each mark is that for 1 mark you have to see about pre and post fertilization, pollination and pollen grains. For 2 mark we have to see diagram based questions on pollen grain and anther, double fertilization, albuminous and non-albuminous seeds, pollination in Valisaria and Nuca and apomixis. And then we have dehiscence of anther, emasculation and bagging. And 3 mark we have to concentrate on pollen bank, pollen pistil interaction, anther, embryo sac and importance of G-seed in angiosperm, types of pollination, outbreeding devices, end product of double fertilization. And then parthenocarpy and parthenogenesis and false fruit and true fruit. And the 4 mark is case study based questions. Okay. So the topics to be covered in this chapter is development of male and female gametophytes. We have pollination, in which types of agencies that aid in pollination and its examples. And then we have outbreeding devices, pollen pistil interaction, double fertilization. Under that we have post fertilization, development of endosperm and embryo, development of seed and formation of fruit. And the 5th one is special modes, that is apomixis, parthenocarpy and polyembryony. And last one is significance of seed dispersal and fruit formation. Let's get into the first question. Okay, it is a diagrammatic question. You have to draw a diagram of Megasporogenesis of an angiosperm. Angiosperm is flowering plants. Okay, they are flowering plants, and you have to label funiculus, micropyle, embryo sac, and nucellus from the diagram. Okay. So here is the diagram. You have this diagram and here you have the funicle. Okay. Funicle is the part where the ovule, this is an ovule. Okay. This is an ovule. So ovule is attached to the ovary. Okay. So this is the funicle. And then you have to mark the micropyle. So micropyle is a small opening over here where the pollen grains enter during fertilization. And then you have to mark nucellus. So nucellus is present in the integuments, which is it is a nutrient providing tissue. Okay. And then we have this embryo sac. So embryo sac, you have to mark it over here, which will turn into a seed. Okay, and then we have other parts, micropylar pole and chalacil pole. So micropylar pole is present near micropyle and chalacil pole is present opposite to the micropyle. Okay. So next question is you have to differentiate between wind pollination, wind pollinated and insect pollinated flowers. You have to give an example of each type. Okay. So we have this wind pollination and insect pollination. So wind pollination is pollen grains are light and non-sticky in wind pollination. Insect pollination, pollen grains are sticky. Why they are non-sticky here? So that they can fly when there is a wind. Okay. Insect pollination. So they are sticky because when a bee or beetle enters into the flower, the pollen grains stick to the bees. Okay. And wind pollination. So when pollination you have well exposed feathery stigma. So this also ensures that when a wind comes, the pollen grains are dispersed far away. Okay. Insect pollination stigma usually present in, between the petals. Okay, in the center of the petals or covered by the petals. It is not like well exposed as in wind pollinated flowers. And then the third point is single ovule in each ovary. So you have this ovary and you have an ovule in it. Okay, so you have an ovule, so it is attached by funicle. Okay, so single ovule in each ovary. Insect pollination, it may be different. So you may have single ovary, single ovule, you may have multiple ovules in each ovary. Okay. When pollination, the flowers are packed into inflorescence. What does inflorescence means? You have a stalk, main stalk over here, and then you have flowers branching out. Okay, you have several flowers branching out of it. So this is inflorescence. Okay, but in insect pollination, they are large, colorful, and fragrant. Okay, when pollinating flowers do not produce nectar, whereas insect pollinating flowers, they produce nectar. So the main example for wind pollinated thing is the corn cob. So you have the corn. So the corn that we eat. Okay, so this is the corn cob. This is the example for when pollinating flowers. Okay. Insect pollination is you have flowers such as hibiscus, etc. The next question is describe the process of Megasporogenesis up to the embryo sac formation in an angiosperm. Or this question can also be written as where does the process of Megasporogenesis start in an angiosperm, and describe the process up to the formation of embryo sac. Okay. So you have to describe Megasporogenesis until embryo sac formation. Okay. So here is the answer to it. So we have Megasporogenesis. Where does Megasporogenesis start? It starts in the ovary. Okay, ovary of the flower. So this ovary will contain ovule. So in the ovule there is structure called megasporocyte. This megasporocyte will develop into an embryo sac. Okay, this megasporocyte will undergo meiosis. So meiosis is a reductional division. So that is there is 2n of chromosomes. Okay, it will get converted into haploid, that is n. Okay, so this is meiosis. Okay, so this meiosis will produce megaspores. How many megaspores it will produce? It will produce 4 megaspores, that is 4 into n. That's what it is written here as 4n. That is 4 number of haploid gametes it will produce. So out of these 4, 3 will degenerate and 1 is functional. So this functional megaspore, so this is a single functional megaspore will undergo mitotic divisions. So when undergoing 2 to 3 mitotic divisions, so this single nucleus only, so you have this nucleus here. So this is the cell. So the cell is here, the cell remains a single cell, okay, and then this nucleus will be divided, so will undergo mitotic division and form 8 megaspore or megaspore with 8 nuclei. So you have to remember here there is 8 nuclei, not 8 cells, okay. So after this you will get 4 travels to the chalazal end, that is opposite of the micropylar end, and 4 travels to the micropylar end. So this is the micropylar end as we saw earlier, okay. And then when it travels, you have 4 in the chalazal end and 4 in the micropylar end as we saw here, okay. And then what happens is 1 nuclei from chalazal end and 1 nuclei from micropylar end will come to the center, okay, and they form the polar nuclei, okay. And let's go to the chalazal end. So chalazal end we have 3 cells, 3 nuclei, and then micropylar end we have 3 nuclei, and they will develop into cells. So now chalazal end will have 3 cells and micropylar end will have 3 cells, okay. So the cells in the chalazal end are called antipodals, okay, and the cells in the micropylar end, there are 2 types of cells in the micropylar end. One is the synergid cell and the center one is the egg cell. What is the role of synergid cell? It will provide nutrients to the egg cell, okay. And we have this antipodals. What does this antipodals do? They provide nutrients to the whole of the embryo sac. So this is the role of the antipodals, okay. So here at last we have this embryo sac. So what does this embryo sac contain? So this embryo sac will have chalazal end and micropylar end. As I said before, it has chalazal end and micropylar end. So at the chalazal end we have 3 cells called as antipodal cells, and the micropylar end will have 3 cells, 2 of which is called synergids and 1 of it is called egg cell, okay. And in the middle we have polar nuclei. So this is a single cell with 2 number of nuclei. So it has 2 into n. So this cell has 2 nuclei, that is of haploid in number, okay. So this is how your embryo sac is formed. So your question is describe the process of megasporogenesis up to embryo sac formation, and for this you have to write, that's a question, where does the process of megasporogenesis start? It starts from megasporocyte. So you have to answer that question. It starts from megasporocyte, which is present in the ovule, and ovule is present in the ovary. So this megasporocyte, how it becomes a single megaspore, and this single megaspore will form an embryo sac. This is the process you have to write, okay. And next is, what is pollen pistil interaction and how it is mediated? So let's go into the answer. So pollen and pistil. So pistil is the female part of the flower, okay. So we have pollen. So there are many types of flowers in the place, okay. So there may be many types of pollen from different flowers and different types of species of trees. Okay, and every pollen they will land on every like stigma. So what is to be done is pistil should have the ability to recognize the pollen. Okay, whether it is of right type or wrong type, whether it is compatible or incompatible. So an apple's pollen will not go and fall on a guava tree's pollen. Okay, so that's how the pistil has to know which is its pollen. Okay, so it is mediated by chemical components of the pollen interacting with the pistil. So when the chemical components interact with pistil, if the pollen is of the right type, it will lead to the double fertilization. If the pollen is of the wrong type, the pollen rejects the pollen by preventing pollen germination. Okay, this is pollen pistil interaction. So you have to write about why pollen pistil reaction is important. Interaction is important. Okay, and then you have to write how does the pistil has the ability to recognize the pollen due to chemical components, and what if it's a right type and what if it's a wrong type. So you have to list these 4 points. Okay, the next question is, how do pollen grains of Vallisneria protect themselves? Or you can also write how pollen grains of Vallisneria have mucilaginous covering. So there are 2 types of question. The answer is the same. So in Vallisneria, Vallisneria is an plant where the male flowers produce pollen grains in the surface of water, and they reach the female flower at the surface of water. So what has to be done? The pollen grains should be waterproof. So what it does to avoid wetting of pollens? They have mucilaginous covering. So you have to write what is Vallisneria and how does the pollens where does the pollens come at the surface of water, and why they have the mucilaginous covering, that is to avoid wetting of pollens. And this is the answer. Let's get into the next question. So this is a case study question. So you are a senior biology student, okay, and you have been asked to demonstrate to students of secondary level in your school the procedures that shall ensure cross-pollination in a hermaphrodite flower. So hermaphrodite flower is a flower that has both male and female parts. Okay, and you have to list the different steps and provide reasons. Okay, so coming to the answer, so you have to first explain why self-pollination is not advisable. Okay, so if a plant undergoes repeated self-pollination, there occurs a breeding depression. So what does this mean? The seeds produced may be infertile, or they may produce, the seeds produced will undergo like give a poor plant production. That's why self-pollination is not advisable. So naturally cross-pollinations occur in plants, but we can have it like man-made also. That is emasculation and bagging. Okay, you have cross-pollination. You mix 2 desired characters of different species of plant, and then by emasculating and bagging. So we have an example here. You have purple and white flower of a pea. Okay, you take a pea plant, you have purple flower and then white flower, and select 2 pea plants. Okay, one with white and other with purple, and you have to label them as a male and female. Okay, so you may label like male as white flower and female as purple flower for this example. Okay, and you have to cut the anthers from the purple flower. So anthers are male part of the flower. because you have labeled purple flower as female, you have to cut the anthers from the purple flower. So this is called as emasculation process. Okay, and you have to cut with the help of scissors, and before they mature or before they dehiscence. Okay, dehiscence is maturing of the pollen and release of the pollen. So before that you have to cut the anthers so that no self-pollination occurs, and cover it with a white paper bag. So this process is called bagging. Okay, so you have to do emasculation and then bagging. And now you have to collect pollens from the white flower, that is male plant. So with the help of a brush. Okay, because white flower you have made it as a male one, and you have to collect pollens from the white flower and then dust the pollens on the stigma of the female flower, that is purple flower. Okay, you have to cover it again with white bag. So white paper bag, why? So that the sunlight can enter. Okay, so cover it again with a paper bag until seed formation. Okay, you have to cover it until seed formation, so that you will get a cross-pollinated seed. The next question is, explain the phenomenon of double fertilization. So we have here double fertilization. Okay, here we have a pollen grain. So this is a pollen. So this is structure of pollen. As you know, pollen will have a vegetative cell. So this is a vegetative cell, and inside the vegetative cell is the generative cell. Okay, so this generative cell will usually divide into two to form two generative cells. Okay, it forms two generative cells. So when this pollen will land on the stigma, so it lands on the stigma, and if it's like the correct combination as we saw before, it forms a pollen tube. So we have a pollen tube here, and it moves through the style. Okay, inside the pollen tube you have two generative cells. The two dots as you see here is one dot, and here is another dot. So these two dots are two generative cells. So if the generative cells can divide to form two generative cells in the pollen tube, or they can be present as two generative cells in the pollen grain itself. Okay, so this pollen tube will travel through the style and will enter through the micropyle. Okay, as we saw in the previous slides, you have this micropyle over here. So you have micropyle over here. So it will enter, the pollen will enter through the micropyle. Okay, and it will enter through the egg cell. So we have two generative cell. Okay, so you have two generative cell. So let me erase this. So in the pollen tube you have two generative cells. So what happens is that one generative cell will go and fertilize the egg cell. Okay, another generative cell will go and fertilize the polar nuclei cell. So as I said before, polar nuclei is two nuclei. So with two nuclei, that is 2 into n. Okay, so egg cell is n. Okay, with one nuclei. So if one generative cell will go and fertilize this, what happens? It forms a 3n. So 3 nuclei. So it forms a triploid. Okay, so it will form a triploid, that is 3n. Okay, and then fertilizing egg cell, what it will form? It forms a diploid, that is 2n. Okay, so you have two types of fertilization, 3n and then 2n. That is why you call it as a double fertilization. Okay. So what does this egg cell after fertilization becomes? So this becomes an zygote, okay, where seed forms. Okay, what this 3n will become? It forms the endosperm. Okay, endosperm is the tissue which provides nutrients to the embryo, okay, or the zygote. So zygote is here, and this is the endosperm. Okay. Now we move back to the question over here. Phenomenon of double fertilization. So you have here this ovule. So this ovule after double fertilization will undergo a seed. Okay, so it forms a seed. Okay, the seed has two parts, that is the embryo and the zygote. So the embryo or the endosperm, and here we have embryo, and embryo is the zygote. Okay. And then we have this endosperm. So endosperm is a nutritive tissue. So we have two types, embryo and endosperm. Embryo is 2n and endosperm is 3n. Okay. So seed is here. Fruit, so it will come from ovary. So ovary develops into fruit. Okay. So this is double fertilization. So what all I have explained is in these points, and we see that here the endosperm is also called as primary endosperm nucleus. So after it fuses, it is a triploid. Okay. So primary endosperm nucleus. So it's called primary endosperm nucleus. So this is the last question, and coming to the summary. After you have completed this chapter, the student will have complete knowledge of sexual reproduction in flowering plants, and main sexual parts of the plant are covered, and about the process of sexual reproduction is also covered, and double fertilization, pollen pistil interaction, and more important terms such as parthenocarpy and parthenogenesis also should be learned in this chapter. So parthenocarpy is the process of development of fruits without fertilization. Okay, so we have parthenocarpy and parthenogenesis. Okay, this is about our institution, and then thank you. Have a nice day.